But yeah, I was asked if I'd just share a little bit about what's going on in the world with selecting genetics for Varroa resistance. This kind of remains the big top issue. Varroa resistance being the ability of bees themselves to fight Varroa on some level. And I just want to warn you right now, this isn't some golden key that means that, well, if you do this, then you can just neglect your bees and they'll be fine. This is what you add to an already rigorous, disciplined beekeeping management process for you and me. It's not something that's just going to fix everything, but it's something that I think is, we're trying to move the bar, having the bees themselves fight Varroa more and more and more, so we can treat a little bit less and less and less. And I think this is the long-term goal. I don't think we're going to get anywhere without this part of the puzzle. And so that's what I'm kind of working on. And I'm just going to throw this out to you, asking if anybody wants to collaborate, because this isn't something you can really do by yourself, especially if you don't have that many hives. And I'm a sideliner, and so there's only so many hives I can handle. This is still my part-time sideline thing. I'm a pastor full-time, a husband and a father, and an adjunct teacher at a college. So I've got enough things going on. I have to figure out how to not let bees take over my life. Some of you are probably trying to figure out how to do that too. But like say, okay, I need to get there once or twice a week and spend just one full day in my bees. And it means if there's anybody that's interested in doing this as well, I'd love to just work together. Here's the three things. You need to figure out how to assess Varroa using integrated pest management, IPM, because you want to treat what's needed, you don't want to treat what's not needed, and you want to see if there's any bees that you have that don't need treatments. Does that make sense? So you gotta keep track of things and analyze things, because those that don't need the treatments are what we want to use to breed from. That's really important. And I think standard practice is, we know Varroa is in there, let's just dump treatments on the colonies all the time, just for insurance. But if you do that, it's hard to figure out now what do we do with selective genetics. So using an integrated pest management process is really important. And then you assess the queens that don't seem to require treatments. How are they gentle or not? Are these ones you want to reproduce? And then you raise the queens from those selected stock. So I really want to just talk about the first two, especially tonight. I don't think we can crunch in a whole course on queen rearing tonight. But it sounds like you're going to do some of that in your field day. You're going to talk about that. So this might be a good kind of intro to that. I think we all know what Varroa are. Just to make sure everybody knows, rarely do you see Varroa on the top of the bees. Usually they are lodged in the underneath plates of the abdomen. And so if you say, oh, I don't see any Varroa, I don't see this, that doesn't mean they're not there. We know the key is they reproduce with the brood. The mother Varroa mite goes into the brood before it's capped, and once it's capped, she lays the male egg, and then she lays females, and they mate together and they reproduce. And so they really spread their diseases in the brood. So our brood are weakened and they vector viruses. And so since they reproduce in the brood, the Varroa population follows how the brood population. So the brood starts getting increase, and then latent from that, the Varroa might start increasing. And so this is straight from Randy Oliver's Scientific Beekeeping website that kind of shows the general curve that we see. If you don't treat or do any interventions, the bee population goes up and down, the Varroa mite goes up and down. And you see in June, you might have 6 mites per bee. But there in end of September, early October, you got 35 mites per bee. These winter bees go in very diseased and compromised, and your colony dies of disease over winter, and you don't know why, because they had enough food, and it's generally often disease vectoring from Varroa. And Varroa mites are easy to see when you peel back, let's say, some drone comb that's, you know, that burr comb between your frames or something like that. It's always, when you see that, you always notice. In this picture, there are actually 3 or 4 mites. I know there's 3 for sure. I think there's probably a 4th. I can't tell if that's a mite or not right here. But so if you actually peel back drone comb and you investigate this, these are the offspring that are developing. And one of the key for us to analyze, are the bees getting rid of Varroa on their own, is seeing if they're keeping this from happening. So you have to have a really good eye of looking for these mites that are in development. These are the offspring of that mother. And that's the key to figuring out, are they reproducing in my hive or not? Are the bees fighting against them? Removing the ones that are reproductive mites. Of course, we know the mites are vectoring diseases like deformed wing virus. So you see a bee with little rope-like little things where their wings are supposed to be. Most likely they're virus-laden because of a high Varroa count in their colony. And so what do I mean by using integrated pest management to assess and treat for these mites? Well, it's kind of 3 steps. So you need to figure out, what's the threshold? What level of mites in my colony can the bees handle and it's not going to destroy them, versus what level do I need to intervene? So you need to establish that threshold, and if it's over that threshold, I need to intervene. If it's not, I don't need to treat. And of course, you need to sample on a regular basis to know, are they growing? Are they staying the same? Are they shrinking? And then the other part of integrated pest management, you can use non-chemical means of control. Try to figure out what can I do in my colonies that would battle against the mites without just throwing treatments in there. And so you're doing that proactively, and then you use chemical treatments only, and the key is only, when Varroa levels are above that threshold. So let's talk about these first 2 items. Mite level thresholds right now. If you take an alcohol wash, you put your alcohol in here, you put your half a cup of bees in there, you swish it around like you're supposed to, and half a cup is approximately 300 bees. 2% of that is 6 mites. So you either take it out and look, or you look from the bottom. Do I have 6 or more mites that fell? That's a 2% infestation. 9 would be 3. I just checked again at the Honeybee Health Coalition website, and this is what they still currently recommend. They haven't updated their recommendation since 2022, but they recommend that your treatment threshold is 2% in the growth stage and 3% in that higher stage. So 2%, I just do April through July, and then 3% August through October. So if I'm that or above in April, May, June, July, I treat. If I'm 3% or above, August, September, October, I treat. And so you're going to learn about this, but here's my way of doing it. You need to take a frame that's by the brood nest. Probably it's good to have brood in it. Make sure the queen is not on this frame. And what I do is I just shake that frame of bees off into a, this is, yeah, it's a Rubbermaid dish pan. And you let the foragers fly away. That's fine, because most of your mites are going to be on your nurse bees. They're interacting with the brood. And I tap them down into a quarter, and this half a cup measuring cup is perfectly contoured to this dish pan. So I get a half a cup of bees. And I already have my alcohol in here. And I dump that half a cup of bees in there. And I close this up. And I shake it up a little bit, but I analyze it later. What I do is I put a little piece of masking tape, and I put the hive number on the top. Because I am batch processing my work. I have to go to my bee yard an hour and a half away, do a whole bunch of work. What I do is I bring them all home and I look at them in the air conditioning back at home. I start analyzing and writing them down. It also just keeping them in there for a couple hours, and they have to ride home in my truck, shaking all the way home in my truck. Like if I look at them a couple hours later, I'm more confident all the mites fell. One of the issues people have is they do the recommendation of swirling for a minute and then looking, and they dump it out. And then sometimes people have said they re-washed it and they found a couple more, you know what I mean? So that's just my way of doing it right now, because it's easy, and I can actually do a lot that way.
No.
Go ahead.
So you do all your hives, though?
Yeah, I do.
I don't know if they use all of that.
Yeah, yeah. So I'm sampling every one of my production colonies.
What's your alcohol wash made of?
Alcohol.
Okay,
I use 91% alcohol. I buy it by the gallon online.
alcohol?
Yep. 91% isopropyl alcohol. I know people say you can use washer fluid, you could do soap. I've tried a few different ones, but alcohol doesn't get rancid over time. If I have 70 mite washes and I want to get to them for a couple days, it's fine. If you do that with soap water, it's nasty. That's why I just stick with it, and you can reuse it, and it does get kind of yellow over time, but I've just found that to work the best. I'm sure there's ways to save money, but I'm about convenience too. So that's how I do it. And so the standard operating mode, if you're a big commercial beekeeper, is you do, let's say you have 500 hives, you do like 50. You know, you do an assessment of some, and then you extrapolate from that what my colony's averages are, and you deal with averages. And if that's the way you have to do it, that's the way you have to do it. But I also know from experience, here's 2 of my hives at my main mating yard. And I did washes, and hive number 1 had 3 mites, and hive number 2 had 27. So I think anybody that says, oh, they'll all just really be really close, I don't believe you. I just don't think that's right. I think maybe the treatment people have something to say about that. They just want you to treat everything or treat nothing, and I only treat some things. So I'm going against, I guess, maybe what often is being told. So what am I going to do? I'm going to treat number 2, and I'm not going to treat number 1. In fact, number 1's a candidate for raising queens from, because this was her 2nd year, and I've never treated hive number 1. So I'm going to go on to a couple other things, non-chemical means of control. I showed this to you, but this I think is the easiest way to get rid of some mites out of your bee colonies. If you want to cut down on how many treatments per year, trap some drones and take them out when they're capped. You've just removed a ton of mites out of your hives. The drone brood, mites gravitate towards the drone brood because the drone is a capped pupa for longer, 4 days, 5 days longer than your worker brood. The mites can reproduce more effectively in drones. They seem to be adapting and know that. And so if I put this in my production hive and I let them cap it like this, that's what it looks like when it's, you got a bunch of drones in there. And if my colonies, if I do a wash and it's got, maybe 5 mites or 4 mites, or, it's close or it's above the threshold, I'm going to take that drone brood out. I'm going to get rid of as many mites as I can that way. If I've got drone in my bee colonies that are in my mating yard and they're keeping mites down to like 1 mite or something like that, I'm leaving the drone brood in. They're raising drones that seem to be a little more effective in fighting against the mites, and I want my yard to be populated by those kinds of drones. So I'm using these in a double purpose way. Removing them when they're way over the threshold, keeping them in my colonies for extra drones for my mating yard, especially in the colonies that seem to be fighting the mites well on their own.
Always putting that in the center.
Usually you put this position 3 or 7, I think, just a few from the edge. They seem to really like that. It's yeah, there I've got it on the center. But yeah, you could put it anywhere in those middle areas. But generally, yeah, generally it's not right there. Generally it's like in position 3 or 7, kind of on the edge of the brood nest. Good question.
One per hive or multiple?
Yeah, I just do 1 per hive. And this is a medium because I have mediums and deeps in my brood nests, but I have deeps as well. So yeah, one per hive. And if they say that they use about 5% of the hive for raising drones, and you have a double 10 frame deep, that's 5% of your comb. So I think it's pretty effective. And other people have said that when you do this, they seem to raise a lot less drones other places. You know, they got it focused on that frame, which, you know, if you do want to make sure you have drones for mating, you are removing that aspect from your apiary, just to let you know. So you have to balance, what do you want to accomplish? But I do know people that do drone capping, and that keeps them down to just having to treat for mites once a year. You know, so it helps enough so treatments are less, because treatments are kind of pricey too. I have club members. I'm the president of our Siouxland Bee Club that meets in Sioux Center. I have members of our club that someone told me he treated 7 times last year for mites, same colony. That's expensive and a lot of work. And so if we're working on looking at resistance for your bees, I think we can get that down to once or twice a year as one benefit. Maybe you can't go treatment free because the mites are still growing, but it's a lot easier to manage bees when you can just treat them once or twice a year than if you have to, oh, nope, they're over the threshold again, and you're just treating all year long. Oxalic during the honey flow and then everything else before and after it, that gets to be pretty challenging. And then finally, use chemicals or your acids, your synthetics, or your organics only when your Varroa is above that threshold when you're sampling. And I do recommend this white paper from Cornell University. It's about 8 pages long and it's a PDF. And it talks about, I think it's the best really quick guide to integrated pest management for Varroa control. And if that's helpful, that'll link you right to it. If anybody wants to just have that link saved in their phone. That's cornell.box.com V and then Varroa IPM guide. That's where you find that PDF and you can print it. The only thing that doesn't have, it doesn't have the most recent oxalic acid treatment option. I think it's called Varroax or something like that. I can't remember. It's a cardboard kind of looking thing that goes between sets around your frames. That's a slow release oxalic. They just came out with it commercially. So yeah, so it just sets like this, just like HopGuard, if you've ever used something like HopGuard, and similar to how some other treatments are. So they're vertical to that. So essentially the approved treatment now, instead of the oxalic sponges, now you have something commercially available that's been approved to treat. Most IPM is described in terms of a pyramid. Why in the world would you talk about that? Well, because at the basic level, you want to do everything you can with the genetics themselves. If you go buy tomato plants, you can buy ones that have some resistance, especially to some of the blights that affect tomatoes. So you look, and this has been bred, so you have a genetic resistance to some pests and pathogens and things like that. That's the golden key. If we could get genetic resistance, that's the foundation. So you start with that. See, what can we do with genetics? And then on top of that, you need to monitor your pests. Then you see, are there any cultural controls? That's like things like, you know, removing drone comb. On top of that, you look for natural chemicals. On top of that, you look for synthetic chemicals. So you want to focus more on the base of the pyramid and then build up from that. And so that's why integrated pest management is always trying to think, are there genetic solutions to this? And that's why I like that model for Varroa treatment. So that was a start. That's number one. It's kind of the basic framework or theory behind that. Now, so what do we do now to assess queens that don't need treatments for Varroa? There's two schools of thought here. One of them has been thoroughly done and demonstrated by Randy Oliver. I call it the Randy Oliver model. And I actually talked to Randy Oliver at IHPA this past November, and I said, hey, I'm using your name, and I want to make sure I don't use your name in vain. So I'm calling something a Randy Oliver model, and I want you to give me permission to say that. And I explained it to him, and he's like, you got it exactly right. That is exactly what I'm doing. Randy Oliver is measuring results. He says, I don't really care how they're fighting Varroa. I care that they're fighting Varroa. And anything that fights Varroa, I'm going to reproduce. Make sense? So it's a results. Randy has devised mechanisms to do mite washes on all 1,000 of his hives. He does that. He's gotten it down to, he says it only takes 90 seconds additional per hive when he's checking them. He's got it down to a really repeatable process. I think I'm about there with how I do it, but I think his solution is probably even cheaper. I don't have enough time and energy to put into it what he does. But anyway, he's measuring results. John Harbo has been around. He's one of the godfathers of Varroa sensitive hygiene and all the research that's gone into that. He's measuring behavior. He's looking at the behavior of bees taking affected pupa that have Varroa reproducing in them and excavating them and getting rid of them from the hive. He's measuring that mechanism, that behavior. And so there's two kinds of ways to do it. I'm trying to do a little bit of both. Like I hear this, I hear that, and I'm like, that's great, that's great. I'm all about trying to understand the best I can what's going on with my bees. So I'm going to talk a little bit about each model. So Randy, again, here's his goal. Regularly test and record Varroa levels in every colony. Raise queens from the colonies that keep Varroa mites to the fewest. And so if he has a colony that's over that treatment threshold, like we said with IPM, he treats the colony and he squeezes the queen and he replaces it with a queen that he's raised from something that didn't need treatments. And he's just doing that over and over and over and over again. And he has a bar graph showing how, 1 year he only required like 5% of his colonies could be treatment free, and the next year it was like 10%, and it was like 20%. He's up to over 50% treatment free. And he's been doing this over year after year, and you can read all about it on his website. If you go to his website, there's Varroa management and breeding resistant bees. And here again, if you want to bookmark that, go for it. But that's Randy Oliver's model, and he could explain that, and you can read all about that on his website. It's I've read everything, and essentially that's what he's doing. What's like, and it's consistent. He goes to hives and he just writes it on the side of the hives: 0, 0, 0 mites, 0 mites, 1, 0, like stuff that's keeping mites like that. Hey, those are the mother queens for the next generation. And the rest of it gets queens from there. So I'm doing some of that. I'm doing that part of it myself. So 2023 is actually, I don't have very good data from 2024 because I was a little bit like a chicken with its head cut off last year because my bees got flooded twice. I made 12 splits in the spring and put them by someone who was begging me, put them by his apple trees, begging me every year. And I said, fine, I'll do it this year. And they got washed away in a flood, and that was in early May. And then the end of June, you probably heard about the Rock River, Rock Valley. I had a dozen colonies, production colonies, right along the Rock River that were 7 feet underwater. And so I wasn't as diligent with this data last year as I have been in the past, but this is 2023. On May 22, I take a mite wash of all 24 of the production colonies I have in my mating yard, and only 2 of the 24 are above the threshold. It's usually that way. In the spring, usually your mite level's pretty low. They've gone through a brood break over winter, and so I treated 2 colonies, I believe, with HopGuard. June 17, I did it again, and this time 17 of the 24 were below the threshold. And the ones that were above, I removed the drone frame, but I didn't do any treatments because they weren't like way above, they were just right at that threshold level. I don't mess with them in July because that's when you're stacking all the boxes on and you got honey on there, and I'm not gonna take every super off every hive to do a mite wash. So then I did mite wash again August 11. And this time 2011, we're still below the threshold. So I had treated 13 colonies with Formic Pro. And then I did a wash in September, and I was back to that 92. So 22 of the 24 are back below threshold. And so, in other words, 46% of my mating yard did not require treatments in 2023. That's after doing this for about 8 years. This is how far I've come. I wish I was farther, but it's where it's at. I'm still doing open mating, so my queens mate with who knows who. I don't have control over this, but just being diligently trying to do the Randy Oliver method over and over and over again, this is where I've come. And I want to encourage you, if you don't do this, just record this in a notebook, stick it in a spreadsheet, and you can kind of see which colonies are doing well or aren't doing well, which ones you might want to flag for saying, hey, that's a good mother for the next generation of queens. And then some of them you're like, that one probably won't live through the winter. And yeah, I know I lost the one that was 12, and it wasn't the 10, it was another one. I lost 2 that winter afterwards. So I had a 90% survival rate of this colony too over the winter. And then I could visualize that. Here's generally what the mite counts looked like in my colonies. So a few of the low ones still went up a little bit. They were still fighting them on their own. They're still just at that threshold, but I'm letting the bees try to fight the mites. And then again, I look at it next year, and the survivors, did they pretty much wipe them out over the brood break over winter? So that's my plan. Here's the question: can anybody actually take mite samples in all colonies? And I say it's hard, but I think there is a way, and this is the best way I've found to do it, is to get enough sample jars or enough mechanisms for sampling so you could do a whole yard. I have 72 of these. I can make a mite wash of 72 colonies and bring them home and analyze them, clean them out, refill them, get them ready to go for the next time I visit a different yard another Saturday. And so, for example, in the spring, I use these double nukes for my overwintering. And in the spring, I move them into these jester boxes so I can either sell them or transport or move them around. So as I'm taking 5 frames of the 8 that are in here overwintered, I'm doing mite washes in that process because I'm touching them, I'm messing with them anyway. I'm just going to dump a half a cup of bees in each mite wash. I got them labeled, so I knew exactly which colonies, because I got them numbered, what the mite level is on each one, and I only sold nukes that were underneath that threshold. So when Andy Joseph came and helped me move all of my, he wanted to, you know, he has to inspect the nukes if you sell nukes. I said, why don't you come the day that I'm going to move them over to Jester boxes and I'll do mite alcohol washes. So he's looking at them, I'm looking at them, and I put one of these on the top of each Jester box. I didn't even bother numbering them, I just put them on top of each one. I only numbered the ones that I needed to treat. And he's like, I've never seen anybody do this before. He's like, you should probably charge another $50 a nuke because you're like making sure they're really healthy. I'm looking at them, he's like, some people are selling nukes full of mites, but not me. So that's the biggest challenge, is actually getting the rigor and the effort of doing all these sampling. It is hard, it's a lot. But if you build it into your plan, once a month, you do a full inspection, which requires washes, you can analyze this and you can keep track of it. The other big model out there is what I call the John Harbo model. And I've talked to John Harbo before as well over a web conference. I never met him in person. And he measures behavior. And so he's testing for Varroa-sensitive hygiene behavior in selected colonies. Then you're raising queens from the colonies that have the best VSH score. And then you want to requeen colonies with daughters from that stock with the Varroa-sensitive hygiene score. So VSH, how many people know what VSH is? Who could explain it at all? Anybody? So this is hygiene. It's bees keeping themselves clean. What is Varroa-sensitive hygiene? Here we go. It's a behavior where capped worker pupa infected by reproducing Varroa, they release a kind of a pheromone, and the adult worker bees uncap and remove that infected pupa. So let's say you got this area and there's 100 infected pupa and 3 of them have Varroa reproducing in the pupa. Varroa-sensitive hygiene bees, the nurse bees that are on that comb, smell there's something wrong going on in that pupa. They uncap it. Yep, it doesn't smell right, and they pull it out of the hive. So they get rid of the bees that the Varroa are reproducing in. Now the disadvantage, of course, that means they're getting rid of some population, but they're getting rid of the population that's infected by Varroa, and they're keeping the Varroa. The result is the bee colony interrupts the reproduction cycle of the mites, and it keeps the colony infestation low. And so that's your score. Is it really, really high? Are they doing this like almost 100% of the time? Or are they doing it never? Or somewhere in between? It's a sliding scale. High Varroa-sensitive hygiene. They're just knocking it out of the park. There's nothing reproducing. No mites in the brood. Low, they're not touching anything. They're not doing it at all. So it's a continuum. John Harbo, you can look his stuff up at Harbo Beeco. He has a link on his website called VSH. It just talks exactly what VSH is. And there's another link called Measure Varroa-sensitive hygiene. And if you click on that link, you can get instructions on how to measure this yourself. And if you're interested in that, again, I'm trying to be in the 21st century here, where people like to just click a little link or grab a link to measure VSH. And what he has essentially is a Harbo assay. It's a test, and it's time consuming. It requires that you haven't treated that colony. So it requires that there has to be some mite pressure. Does that make sense? They have to be dealing with mites and getting rid of mites. You can have all kinds of, hey, I've got a great Harbo assay because I just treated and there's no mites in there. So what you do is you remove 100 pupa that are really close to being emerging, 8-9 days post-capping. In other words, you let the Varroa have maximum time to reproduce under that pupa. So you take a forceps and you pull them out and you look for reproductive mites. Are there any reproductive mites in that cell? Are there any reproductive mites on that pupa? And you do that 100 times. And if you don't get any, if it's 0, you do it 100 times more. A Harbo score is based on the number of reproductive mites per 100 or 200 pupa. Here's what it looks like on the back of my truck. I'm sitting there, I use a grafting frame. I'm taking mites out and I'm putting in piles of 5 of pupa out here. And I'm looking with a light and some glasses to see if there's any reproducing. This is a emerging frame, right? They're hatching. They're pretty old. They're about ready to emerge. They're even brownish. I'm giving them the max amount of time to reproduce, and I'm doing these assays. And here, this was an example of a colony that had a perfect Harbo score. I did 200 pupa, and there was no reproductive mites in 200 of them. Every now and then you'll find one that wasn't reproductive. There's just a mite, but for some reason she's sterile. She didn't have any offspring. And that doesn't count against it. That means, there's nothing reproducing in there. I don't need to get rid of that worker brood. And so that's, the Harbo way is another kind of just as much effort, right? If you're going to put a whole ton of effort into sampling everything, this is a whole ton of effort into analyzing your colonies that you think might be very...
How long does it take you to do?
Oh, like 15 minutes or so. 15 to 20 minutes per colony. Yep. I used to think that you had to have a lab for this kind of thing, that this was like, and then I learned that anybody can do this if they want to. So yeah, maybe longer the first time you do it, but I can do about 100 every 15 minutes. So maybe a half hour for a whole. If you have, let's say 2 or 3 mites in that 100, you know you're not getting a perfect score, so you only do the 100. It's like a score of 0, 1, 2, 3, or 4 is how the Harbo scale goes. Another hand? Yeah.
Okay, now you're able to see these mites with a magnifying glass on these?
Yeah. Yep, just a headlamp. If you have a headlamp and you're looking into the cell, there's always going to be some little excrement, and there's going to be these little white mites either in the cell or on the bee.
Are they moving? Can you see them actually moving?
Yeah, sometimes they're moving, sometimes they're not. But remember those midges that we saw earlier, that when that drone, yeah, those little white ones. So you're looking in the cell for offspring and a mother. And if you see nothing, okay, done, next one. You just examine every single one until you find mites that are reproducing. So generally what John Harbo is in his research is if you have mite pressure, you know there are mites in there somewhere to be gotten rid of, and you've got 200 pupa that are clean, that's a very hygienic colony. What also is going on, it seems like in Varroa sensitive hygiene colonies, is there's a lot, often it seems like bees are more likely to uncap and then recap. They're investigating a lot of cells like this. When I started off, I was told that's bulb brood, that's bad. And I guess if you were to have like a big line of them, you know, maybe that means there's something going on, like a wax moth digging underneath the pupa or something like that. But if it's scattered like this, then it seems like the bees are investigating. Right now, I look at that and say, awesome. It looks like they're hygienic. They're inspecting pupa, and they're uncapping and recapping. And right now, that's kind of the edge of the research, is people are trying to figure out, is this disrupting Varroa as well? In other words, is what's going on in these ones they're uncapping? I mean, we've looked at them and you can pull them out and there's no Varroa in there. I don't know why they're uncapping. It just seems like hygienic bees are they're just investigation, like, I think I might smell something not quite right. I'm going to check it out. Oh, all right, cap it back up. And so I'm just saying, if you see any of that in your colonies too, don't be alarmed. It could be a sense of, a sign of hygiene, and just be just fine. So, yeah.
As I listening to this, you're really looking at the genetics of the queen to determine if they're resistant. Has anyone done the same thing with the drones? That way there you have both a male and a female that are resistant, and then you have a stronger hive and
Yeah.
a stronger resistant. Has anyone have they done the same thing with drones as with queens?
Well, what we're looking at is a colony, and this colony is all offspring of this queen. Drones and workers are the offspring of that queen.
Yeah, but when the queen that has been produced flies off and breeds, you have no guarantee that
Right.
it's going to breed with a drone that is.
Right. Mm-hmm. Yeah, so drones that are offspring of a hygienic queen are going to pass on some of those hygiene genetics to the other queens.
But there
Yes.
hasn't been any tracking of that.
Yes,
There has been.
there has, and that's why your most hygienic colonies are artificial inseminations. So that's why a lot of people are doing instrumental insemination of queens. They're taking drones, let's say they've got 20 colonies that are just scoring top on all the hygiene. They take drones from those colonies, squeeze them, harvest the sperm out of them, instrumental inseminate those into queens. Now you've got both sets, and those are often sold for $1,000 as breeder queens that now you can take their offspring from. Yep. So I'm not doing any of that, but that's going on in the industry, definitely. So Corey Stevens in Missouri is one of the leading people that's doing this work, and he does instrumental insemination of queens with his highest scoring colonies, and he sells breeder queens for you to buy.
Are there any
Yep.
variety of bees that have a higher potential of this resistance than other varieties?
Some Russians seem to have this more common inherently, and the theory is the Russians have dealt with Varroa longer. But you can add this as an additive trait to any bee. So you can get Varroa sensitive hygiene Italians, Varroa sensitive hygiene Carniolans, it's a trait, not a breed. And by having some bees that have this trait and then breeding them with your bees, they gain this trait more and more. Now it's true that, like, when I have a queen and she goes and gets mated, some of her daughters will be really high and some of them are low. Like it kind of depends on probably drones and other things. You know, that's how genetics works. So you don't pass identical things onto all your kids. And so I'm kind of working on trying to figure that out. What's generation 2, 3, 4? How much do I lose? How much do we retain of this? That's still going on right now, the research. So does that make sense? Those kind of two ways of going at it. There's a third thing that's come out. And it's another way of measuring behavior. But Kiara Wagoner with University of North Carolina came out with what's called a UBeeO test. And UBeeO stands for unhealthy brood odor. What she did is she analyzed these pheromones. What kind of smell do these unhealthy brood produce that triggers this behavior in the nurse bees and worker bees? And she worked with chemists and, like, what's the difference between a brood odor, and put that on a spectrum, and then an unhealthy brood and put that on a spectrum, and saw these couple little spikes where it was different. And then they synthesized that pheromone and produced it in a spray form. So now what you can do is you can take this synthesized pheromone, get a little PVC. I was looking for, I have a kit and I was looking for it. I was going to bring it, but I couldn't find it before I had to leave. So you have an applicator and you spray some of that pheromone on a select set of cells, and then you put it back in your hive and you wait 2 hours and you take it back out and see if they've uncapped anything. It's another test of that behavior. So this is an example of what you would say, this is a score of 0. They haven't really uncapped anything. Or this is, you know, what would it look like if they didn't touch anything? And this is almost 100% because they've checked everything. And so it looks like this. It comes from Aptera. It's an applicator, and then you get these vials, and it's really expensive right now. I think I spent $500 on this last year. But here's a photo of me doing it. I just grabbed this frame out of my hives, and I stuck that little PVC thing there, and I marked on the top where it was so I could go back easily. And you do 3 sprays on that, and this kit comes with a little tiny, it's a little tiny whiteboard. So you just put with your marker the date and the colony ID and the time that you started the test. You put it back in there. You wait 2 hours, and then 2 hours later, and this was 7/19, so a little bit over 2 hours. I pulled this back out, same date, and you measure how many they uncap. And then you give it a score, a percentage. So that's kind of the leading edge of ability to test these and analyze this. So here's an example of a colony that scored 0. Put it on there. You kind of barely see the indent here where it was. And it's good to have like a couple cells for reference, you know. Those were those that were open. So that was one I didn't expect really high because I had to treat this before, but I just wanted to try it out. Here's an example of one that scored really, really, really high. In fact, I learned after that this can sometimes stimulate them to hatch early. So you see some of them are coming out of their cells. Well, I got one. I guess I should have known that. It looked like one was emerging. Maybe even 2 were. But anyway, the bees, my score, 96% of the cells that were sprayed there were investigated. That became a breeder queen. Does that make sense? So, and I've got tons of photos of this because I am working with a guy who's treatment free in Vermont. He's sending me queens, and part of my payment is I do this score and I send him back the results. So he's like, I could spend a whole ton of money on UBeeO testing. If you've already invested in it, why don't you send me back the results? And then we're trying to get to the point where, yeah, if some of the highest scores, I'll send them back. So I'll just use them for breeder queens. So I've tested at least 30 colonies in 2024 using this newer method. And so again, once you kind of select down what you want, you raise the queens from the selected stock. So I'm looking for gentle, productive, and Varroa resistant. I overwinter everything here, so I only raise queens that have lasted 2 winters here. That's kind of they start being built in a nuke in July or June. Year 1, year 2, they're a production colony. If year 3, they're still around. They haven't swarmed. I haven't needed to treat them. That's who I use to raise my next generation of queens. And I'm just in that cycle, trying to move the bar as much as I can against mite-resistant bees. And then of course, I'm also populating my mating yard with drones from colonies that share those traits. And so I've got 3 big yards, and sometimes I'll take a colony that's really high scoring in that yard and I'll bring it to my mating yard. And I'll switch them around. So I'm just trying to keep bringing the best scorers back to the mating yard so they mate with the queens, and then I bring them back out there and test. And so I'm requeening colonies from time to time in that way too, but that's just my way to try to maximize genetic diversity with all the bees that have the most desirable traits. And that's the life of someone that's trying to raise Varroa resistant queens. By the way, there's a difference, I think, between resistance and tolerance. This is another area of research that's happening right now. Some colonies are good at keeping their numbers down. Some colonies seem to be good at surviving even when the numbers are high. And that's a valid research area. I'm not doing that. I'm not looking for tolerance. I'm not letting my Varroa grow too high. I'm looking at resistance. But there are people that are also at the same time trying to figure out what is it about bee colonies that seem to survive even when the mites are a higher load. That's just not my niche. I'm in my lane. I'm trying to learn all of that I can in my little lane, but I want to acknowledge that that's also something that's going on in the research. So back to this question. Anybody want to collaborate on this? Get in the process of using IPM, and then I'm willing to even help assess the stuff that doesn't need treatments. I'm gonna invest some more in UBeeO. Got a couple people in the state from IHPA that want to work together, so I might be driving to some other yards and doing some Harbo assays and UBeeO tests and trying to, oh yeah, this is good. I'll bring your queen back with me. I'll give you a couple of mine. You know, just kind of helping each other out. Because if there's anything that I know, we need to work together in this. Yeah. We need to support each other. So that's my offer, and I hope that, yeah, I think everybody that's got a decent sized yard, there's a chance that you've got some resistant bees, and if we know about them, we can focus on reproducing them as well.
All right, we're going to get started here. Welcome everybody. We got some new faces here. So my name is Richard Vondero, president of North Iowa Beekeepers Club. That's Eric Folmuth back here. He's our secretary. He's video recording so we can post all this stuff. So everybody got a chance to, our door prizes are over there. Got a few things on a table over there. Anybody can bring anything for a door prize. Doesn't necessarily have to be bee related. It's kind of just a little gift that you can get or put a name in. And our raffle drawing is our easy check, Mite check back there for this month. So we have different things every month, and this month we have that. So $1 a chance for that. That $1 goes to help offset the cost of not having membership dues. So it's kind of a way to raise a little money for a club and not having dues. So we'll get started here. So I laid back there, I don't know if you want to, list of our 2025 schedules back here. So we have one more. We have our May 17th one here. I don't think that's right. I think it's the 8th. It's the second Tuesday, the 8th, actually.
What month? May?
April. April one is
8th. It says April 8th.
April 8th. Okay, there's an April. Okay, yep. So, yep.
Don't have one for May because we
We got the field day, spring field day, yeah. So the ones in the red, we have our June 3rd. It's not the second Tuesday of the month, it's the first Tuesday of the month because the second Tuesday I'm on vacation. So the June 3rd, July 8th, and September 9th. That's our meetings that we go out to people's places and look at their bees. So it's a chance to do, it's more of a hands-on. We get to go into your beehives. We got yours last year, wasn't it?
No.
Was it 2 years ago?
No.
1 year? Okay. But we get a chance to go into people's beehives and see what they look like and stuff like that, and ask questions and help them out to, hopefully, get them through the year. So those are our 3 summer ones that we do kind of more of in person out there. So we have our Spring Field Day one, May 17th. That's down at Pat and Peggy's down at Goodell. So it's a full day down there. In the morning we're going to talk about different ways of raising queens. We're going to do grafting queens. You get a chance to actually graft if you like. And there's different ways of actually raising queens. So that's our morning. The noon is we have a potluck lunch, so everybody brings something. We have a lunch. And then in the afternoon, we go out to beehives at Pat's, and we do like 3 stations out there. We have, Pat will do like splits, how to make splits, how Pat does it. We have hive inspection, and we actually do the mite count, how to do a mite count on your hives. So you get to learn firsthand what frames you got to look for and how to do it actually. So, but it's a full day, $15 a person. So I have registration forms back there. We're going to actually post it on our Facebook page too, so you can just pull them off there if you had to too. So that's up there. We have also our club auction here down at Pat and Peggy's again. This is April 26th. It's a Saturday. So we usually have a lot of good stuff down there. Pat, do you got any more 2-3 consigners so far?
I got a couple so far, yeah.
So far, okay. It's early yet, so I got a few things I'm going to put on there. So if you got anything that you want to, don't need anymore, put on an auction, get a hold of Pat. Yep, turkeys are out there watching us. So get a hold of Pat and we'll get it consigned. If you're looking for equipment, it's a good place to find some used equipment. And we'll post when we get closer what's actually been consigned so you get a chance to look at it and know what's actually there. So the other one here is, this is through NIAC out there. It's our, Pat and I just finished our beginning beekeeping class out at NIAC here just last week. And so, but we have a summer one. So the summer one, you actually just go through NIAC and you register through NIAC. It's $89 for the entire four-week session, or it's $25 per session if you just want to pick and choose which one. So the first one is like May 10th. It's basically starting right, installing nukes and packages. We'll have bees out there. So everything we do out there is in the beehives. We start out in the class for about an hour, then we go to the beehives. So hopefully we should have one that we actually will be making a split off of too yet out there. So we'll see how it goes. May 31st is basically at hive inspection, what we're seeing, how to grow it, how to get into, starting to, get it built up for honey production. July 12th, we're actually talking about honey production and bee diseases. And then the last one is September 13th, basically prepping for winter and keeping your bees alive. So that's the NIAC class out there. I have a phone number on top. You just call NIAC and get you registered, and I'll get you set up. So that's on there. On other things we have here, March 26th, here coming up is Iowa Honeybee Day. Down at the actually state capitol, we go down there. I believe it's a Wednesday if I remember correctly. It starts at 7 o'clock. They let us in at 6:30. We can get in the door. Iowa honey producers put this on. It's a way to go down to talk to legislators about different things. One of the biggest things we've been pushing for 3 or 4 years now, is to get the honeybees as considered livestock, so we don't have to pay taxes on selling bees. So that's one big push we're really doing. Hopefully it should go, hopefully this year maybe we can get it to go through, but we got to talk to legislators about why we need that. So it's a good day to talk to a lot of different people.
So that's 6:30 in the morning.
6:30 in the morning they let us in, yep. And then it goes to a few hours until like 11, but some of that is we get to actually probably do a little tour too yet sometimes. Sometimes they have one tour, you go into the agriculture department main office. We've had actually a second tour where we've been clear all through the Capitol, been up close and up in the dome too before. So you get a couple things there, but March 26th. And okay, let me see. I gotta check this. Oh, Central Iowa Beekeepers, their spring field day. That's March 15th down there. I know they have a registration form out. If you go to Central Iowa Beekeepers Association, there's a registration form out there. They have a full day of speakers down there. A lot of good information down there too yet. And that's held down at the Arboretum and Gardens in Madrid, Iowa. So I have an address here too if anybody's interested in it, then we can get you hooked up with that. So, of course, our auction is April 26th. SEBA also has an auction, Central Iowa Beekeepers. Theirs is now May 3rd. So there's their auction down there. And that's held down by